Hey there, let's talk about the following part and last video for that chapter 13 about evidences of evolution. So we'll talk about anything from Pangea to biogeography, homology, analogy, vestigial trait, and then we'll look a little bit at how we map this on trees and how this matches with our barcoding project. So let's dive into it, shall we? So here, if we focus on this picture here of the Pangea and where we found fossils, you notice how the distribution of those fossils on the Pangea is like, I should say that differently. The distribution of the fossils in the world matches the Pangea of 250 million years ago. Notice that all of those are after, so all the Mesozoic, those are Triassic, Permian, Triassic, all of those are from the Mesozoic, so the age of reptile. That was when Pangea was one. And so when you look at this, we find fossils that are similar, same species from South America and Africa. However, nowadays, there is a giant ocean in between the two. And the only way to explain this is to rely on the theory of plate tectonic, which states that 250 million years ago, Pangea was one continent, but little by little separated, right? So it makes sense now to find fossils from the same time period in South America and Africa, or in Africa, India, and Antarctica, only due to the movement of the plates, right? The induction of magma underneath by the core of iron rotating and the magma being super hot, and Earth is cooling down through time. It's fascinating. If you are into geology, I would encourage you to dig into it. For biology, we only care about the consequences of the plate tectonics. So this shows you how you can find fossils that are at different places, but yet that are similar species and from the same time, right? So that explains that the distribution of species was that species population here was all across the two continents. It's important also to note uh, that those fossils show similar adaptation morphology, right? So it would say that those fossils are from a continuous population, right? With that being said, we also have now the Pangea explains something really interesting. So I would drive your attention towards like the Indo-Pacific, Indo-Australia, the Indonesia region over there. If you go back, you notice that India and Australia were completely together, right? And they've been separated here between. So in Pangea, it was together, and then they started to be separated. India, the plate of India moved the world apart here. So you have a separation that was quite a long time ago, 150 million years ago. And it means that after when the land came out here, Indonesia came out, species started to colonize, populations started to colonize from either side, right? And that explains why nowadays we find this extremely atypical line that we don't usually observe in the populations. How from one side of the Wallace line, the sign towards India and Asia, you have species that are really like Asian species, whereas from the side towards Australia, we have species that are way more like Australian species. And it's really explained by that migration of India away from Australia after Pangea started to break apart, right? So this line is extremely striking, and it's explained by the plate tectonics, by evolution, and we call this study the biogeography. It's a study of how geography informs biology and vice versa. So if you're interested in that type of study, there are plenty of work out there to be done in biogeography, and it's really fascinating. It relies on geology, biology, geography, so it's a really cool area of study, and it's not only about plate tectonics, but it's using information about geography to inform biology. And we'll talk a little bit more about this in class or in the next videos. Okay, so now that we have an understanding of this, I wanted to talk about since we need the distribution of traits, we also notice that there are some similarities for those traits across species. So if you look here, it's color coded. Each bone that is similar is color coded, and the fact that those bones are all doing the same job regardless of the outward appearance of a population of an organism. We call this homologies. Homologies, logi similarity, homo from same descent. So it means that those traits are shared between those organisms due to coming from a common descent ancestor. It's not likely that each of those organisms populations have evolved that arrangement of bones independently. It's way more likely that they all got it from a common ancestor. So it tells us that the body plan of having those bones in arms, limbs, is way older than any of those species, right? So it tells us that our ancestors were tetrapod, meaning they have like 4 limbs, right? And it's the same for all of those organisms. Now, if we do the flip side of this, we can find analogies, which are structures that are similar but not due to common descent. You see a bird has wings, a butterfly has wings. It's not because wings was an ancestral trait, it's because they both evolved. Population of hawks and moths and butterflies have evolved similar adaptation, flying independently. Why can we say it was independent? It turns out that when you look at the tree of those traits and how they arose, you can find out that if you get an explanation as common descent, which means that there's an ancestral trait of wing, it means it would require so much evidence of losing wings that it's not parsimonious to think that there was an ancestor that could fly to all animals that are in common between the insect, butterfly, and the hawk, which would mean basically all reptiles, mammals, basically everything but invertebrate almost. It would mean that that's a common ancestor, including invertebrates, of course, like butterflies are invertebrates. So it would mean like just maybe sponges and plants that are not involved, in that common ancestry. And it would mean so many more events of losing wings, right? So it's not what we call parsimonious. So when you look at a tree, we go with the most parsimonious explanation. It's way more parsimonious. It requires less explanation power to say that wings arose here and there in the tree, and then maybe were lost for vestigial trait. We'll talk about it in a second. Rather than saying it was an ancestral and then it was lost for C, it was lost then for E, right? This is a simplified case, but if you look at the tree of life for all life forms, it is way more parsimonious to say that this is an analogous structure than an homologous structure. So that's also how we decide homologous, analogous, And finally, the vestigial structure here in the middle. Those vestigial structure, they are animals that we would expect by homologous descent to have the trait, but they don't have it anymore. So think of the hind limbs of whales. They are coming from tetrapods, and they are cephalopods, so they're supposedly having 4 limbs, and yet we see only 2. And it's because there's a vestigial structure. It's still there. So is in the snakes. You can still see the structure. It's just not developed anymore. So it's not that they lost it. It's more that it's vestigial in the sense that it's underdeveloped because it's not necessary anymore. The population has evolved different traits. And so for that, the evolution can also be selecting for loss of those traits. So how do you think about vestigial traits in terms of evolutionary terms? Well, think about it that way. Any organism in the whale population who are still developing hind limbs, they invest resources in that, right? And so those resources are taken away from the rest, including reproduction. Thus, they are losing differential reproductive success, but they don't gain anything from having those limbs. If anything, they're even losing hydrodynamism, right, in the swimming position. So when you realize that in the ancestral population of whales that had 4 limbs, any organism that was born with random mutation leading to ever so slightly smaller back limbs, hind limbs, were an advantage in water. And thus, the hind limbs were selected against. So it's again thinking about that evolution in the sense of not selecting for the fittest, but more selected against the lowest, the less adapted organism, right? Same thing for the moles. The moles are blind. They don't have an eye. It's a vestigial eye. Any mole that's born with an eye, it's just not really adapted because they get into dirt. It doesn't really help them. So you expend resources for things that's not good for your survival and your production. Most started to have, and they had highs initially. They lost it because it was more advantageous to, you were able to put more resources into other things than into production, essentially, than into making that high. We're talking about the timeline of millions of years, remember? So we're talking about slight differences that compound through time via selection, right? Remember, variation of a trait, irritability of a trait, differential reproductive success for the trait, due to the trait. And finally here, how does that match for our barcoding project? Well, if we look at mutation, so we can look at actual DNA and infer the same thing about homology and analogy, but we can also just simply look at the rate of mutation. So if you have a common ancestor with a sequence that we found in a remain, and then we assume that two species, two populations evolve independently, they start to accumulate random mutation, right? Those random mutations occur at a rate of every 25 million years for that particular organism. We know that due to reasons we don't dive into this class. Well, then it means that two nucleotides have changed for the modern species 1, and two other nucleotides, really likely other nucleotides, would have changed for modern species 2. So when we have 50 million years of common species of a common ancestor of two species, and the rate of mutation is 25, we expect 4 nucleotides to be different, because 2 and 2 is independent, right? That will, if it is on the coding region, lead to speciation, but if it's in a non-coding region, there could be speciation for other reasons, but if those mutations are non-coding region, aka introns, that can be used for barcoding. This is how the barcoding happens, it's accumulation of random mutation, right? This is also used to make trees, and we learn that later in this class. We can use this rather than the traits, we can use DNA to make trees based on the number of nucleotide changes. So there are a lot of things that you don't know about how to make that happen. We need to be able to estimate the mutation rate. We need to be able to estimate the coding or non-coding nature of a region. And we need to have the DNA of the ancestor to compare to the modern species, right? So all of those factors have to be compounded, but overall, we end up having a better vision of evolution thanks to biogeography, ancestry, so analogous trait from common descent, homologous trait, sorry, from common descent, analogous trait, but are just similar functions, but not coming from common descent, coming from independently evolving populations towards a self-solution to a problem. And then vestigial trait, when traits have lost their benefit to reproduction and thus are selected against, but you still need the variation, right? So there could be many traits that are not good for survival of an organism, and yet the population doesn't see an alpha frequency change because you just don't have variation. Everybody has the trait, nobody's born randomly by chance without it. So it requires this, right? Don't forget that. We build then trees using the parsimony principle when we have to choose between an ancestor homologous trait or an analogous trait based on how many changes will we need to invoke to explain what we see today. And finally, the barcoding project relies on those independent mutations being accumulated into the introns of a coding sequence. Okay, we'll stop there, and I'll see you in the next video. You guys take care. Bye-bye.